Hello friends, welcome to lecture 3 of week 3. In this lecture we will look at what is R and how it is used in various genomic and statistical analysis. So like we have seen Linux as a very important tool and interface and OS which is used to basically run all the programs that you will be using, making scripts, right. Similarly, Perl and Python are the good scripting languages for any genomic or metagenomic or basically biology related compression analysis. And at the same time, R occupies a very important position because it is a programming language which is developed for statistical purpose, right. So, statistical computing and also making the graphs and plots that you see in various publications. So, this was developed by statisticians Ross and Robert, right, and it is freely available. There is something called MATLAB also, but that is more comprehensive and commercial, and you have to get license for that. But R is free, and so many people all over the world are contributing to R by writing their own programs and freely available programs which you can use, and also you can modify. It's an open source. So it is one of the most commonly used programming languages in data mining today, and perhaps like the most widely used in the area of biological sciences, and you should know R if you want to do the data analysis and also for the statistical analysis mainly. Okay, so basically used by data miners, bioinformatician, statistician, for data analysis, for statistics, right. And there are so many tools and additional software which are available developed by people that you can find published versions also in GitHub and also in the R library which you can directly use and download. Okay, so basically R can be command line also, like okay, so you can download the command line version, or you can also have a GUI based version which is called the R Studio, which is very good and easy to use, which gives you a nice interface of you can write your code, manage data and files, and also delete files and make plots. So basically R Studio may be more convenient, and it is also free and open source, which is important. Okay. So to download R, basically if you just search R statistical programming suite in Google, you will know, but basically it is available here, and also the page looks like that. You will have options to download for Linux, Mac or Windows. Okay, so which are the common platforms, and for all of these it will be free, right. You can see that what are the latest releases, as we have seen earlier in the Linux. It is available, for example, in tar gz format, and you can easily untar and gunzip and untar it and then install it, right. And you should also look at what version you are using, because many times the version issues may come that it may not be compatible with other languages. So that sometimes is an issue, but the most updated version you can avoid and go to the last version. Okay. So yeah, so this is the page from which you can download R. Okay, and now this is the GUI that you will get in R Studio. Okay, so R Studio is a GUI based platform for using R, and which is very convenient. So it provides you like a console that in which you can write. For example, there is a command prompt here where you can write your code. Okay, you can use the common things like if you want to see the demo for some of the software, or help, or basically to quit R, so you can use these like Q bracket. So basically this is the convention in which you use the R various options, which you will see later also today. Okay, so what you have done is will be here. Okay, your environment. So sometimes the outcomes of the console, okay, will be saved here, and your files and other thing that you have made will be here. So okay, so basically your history kind of thing will be here, and you can always delete. We will see later. So basically you can see that this is a comprehensive console where you can which you can use for the statistical analysis. So like that was in white color, this is in black color, and this you can always specify that how you want to view the console. Okay, so same way like the history or the environment panel, your console, where actually you will be, you will be seeing that what you are doing, right? And here you will execute and write your code, and it will appear here, right? So now since it's a statistical and mathematical operations are allowed here, so for example, if I just type 1 plus 2 here, it will give me the answer 3. Okay, so that means it is working like a calculator, and because the objective is that you can use it for mathematical and statistical operations, and also you can take the output that you are getting from any such expression or calculation into a variable. And defining the variable is like, you don't need any kind of predefined, or like in C you have to define as an int or char. So here you can directly say that okay, this is the variable, and whatever is the outcome of this or output of this should go into this variable. And now this variable, if you type, it will give you the answer 3. So that means the outcome of the first calculation was saved in a variable. Okay, so this is how, like this is a very simple example, but the complex programs and scripts also will be using the same convention. Okay, so basically, so for example, here we did this calculation. Okay, it was stored in this. So now this is also available in your environment or history file, that there is a variable calculation that you result, and the value was 3. Okay, so and whatever you have done earlier and the files and in your current directory will be here. Okay, like what files you are using, the file size. So basically your current working directory, okay, is, which you can set, we will see in the next slides, right, is here. So you can use that, or also drag drop for running a code, and the dates and these things that you have created those files. Then when you are using it, there will be several files or things that you want to delete. So there is an option of this broomstick, or like, okay, which you can use to delete any particular selected file or entire thing which is there in the environment or history. For example, from this list window, so you can see that okay, you want to use it as a list. So right now it is as a list or in a grid view. Okay, and then you can select that what you want to delete, and just use this broomstick. Okay, so this will delete the previous files and things, so that you don't get confused, and also for your optimized usage of the space. Now to use R functions is not difficult. So like for example, you just want to know what is your current working directory. In Linux also we have the command pwd, present working directory. Similarly, get wd and brackets. So this will tell you, like in the prompt you can write, this will tell you what is your current working directory. Okay, so like for example, this is right now shown as a, like in a Windows C user Lenovo, like that. Okay, or it could be so okay, so now and similarly, like you can also set, and so your working directory is the one which is shown here. Okay, so you would like to have the directory where all your files and input material is there for running any code, or okay, right, so that you don't have to define the path. So you can set any working directory. We will see this. You want to install any package that is available in the R library, you can install just by install.packages, and in bracket you can define that package. Okay, and similarly, like this bracket means that if you leave it empty, it will tell you the default. Otherwise you can specify something within this brackets. Okay, then loading any library, making a plot, you can put anything in this bracket. Okay, reading a table, which table, and which or a CSV file, right. So for example, you want to know what directory you are in, just say get wd this brackets, and it will tell you that this is your directory. Now you want to set another directory, so you can say like set wd and define the directory path here. Okay, so now your directory you have set to the, basically you have moved to now this directory. Okay, right, and now when you say again get wd, it will tell you the directory that you have set. So now in that slide window you will see your files that which are there in that folder for the analysis. Similarly, like you would be installing several packages, because to use a package you have to first install it. Okay, and since your system is connected to the internet, it can directly get the packages from the source and install it. So like install.packages, and one package is here. Okay. Similarly, you want to install multiple packages together, so just in single quotes you can define and separated by comma, so it will install all the 3 packages together. Okay, then basically from the commonly used R packages, this tidyverse is one package that contains a lot of tools such as ggplot, dplyr, tidyr, readr. So basically, which are the common ones that you will be using for R and various features of R, and commonly used in like biological science or data science, because you would like to do this statistical test and make a plot or a figure, so that you can know that what is the outcome of that, and you can the values are important, and also the plot, because you want to report that in your paper. So ggplot is one of the application that can do. Okay, so again, like to install, you can install each of these separately, or you can just say install packages tidyverse, so it will get all these packages together, it will install. Okay, so now running a code is okay. So basically, you can create a new file from the R notebook. From the top, you can say file, new file, and R notebook, and or anything, any name that you want to give. Okay, and then basically you can run any program. Okay, for example, this is two commands you are running, plot this and plot this. Okay, or you can also separate that by this way and run either of the code or the chunk. Okay, so basically, or you can also copy it from somewhere, or just paste it directly, Control Alt and insert I. Okay, so now if you want to run, you can select that package and just say run from here, it will run that particular thing. Or you can also have the commands to run the entire thing, like run all which are available in that console, or run all chunks above, or run all chunks below, like that. You can run any code in R. Okay, and the file that you are basically the outcome will be saved as a .rmd file. Okay, or basically which is a NBHTML file, and it can be HTML, and also basically you can suitable for display on a website. Okay, that is why it is like HTML format. Okay, and it can be viewed in any common browser such as Google, Chrome, or Firefox, etc. And also it can be opened in the R Studio, because there you would like to see the outcome, or do for some further analysis on that file. Okay, and so basically you can always create projects, and in that also you can share save that. Okay, now ggtree is one of the commonly used R packages to visualize and annotation of phylogenetic trees. Okay, so like phylogenetic analysis we will be doing in the future classes. So it is the analysis in which we want to look at the relationship between a group of sequences or organisms which are okay. So basically which are represented by sequences. You might have done a multiple alignment before, and the data that you have, you are using that outcome of the data that you get from multiple alignment, or some other analysis into the ggtree. Okay, so basically Newick is one of the outcome format of the sequence alignment or the tree. Okay, so you can get the packages from, say, bioconductor also provides you a lot of packages for which you can use for R. Okay, so ggtree usage is again like simple. You have to first load the libraries like tree.io and ggtree. Okay, then this is your data which is in the Newick format, sample.newick. Okay, and packages like tree.io which you want to use for drawing the tree, and the entire thing you are saving in a variable nwk. Okay, then basically you are reading this Newick tree in the tree, and then you are plotting the tree using ggplot tree and defining the other parameters, so that you can draw multiple types of tree. Okay, so basically it is a very good application for drawing the phylogenetic tree. You can also do much more, like naming particular or coloring, or naming the tree, coloring the tree, like that with the ggtree. So this is the most basic visualization. Okay, then again, like there is one more package, edphilo, which is used for the comparative analysis of the evolutionary data that you have. Okay, So basically orthologs, that also you will be covering in the later classes. So orthologs is one of the outcome of the comparative analysis, where you want to see the genes which are commonly present across organisms, right? So there could be a group of related or distant organisms, but there could be genes which are commonly present across all of them, and which you have found as orthologs using some parameters. So basically you want to quantify and describe the phylogenetic signal which is coming from those orthologous gene sets from multiple species. So this can help you in that analysis. Okay, so for any package, this kind of page will appear where you can see that this is the package name, what it does, okay, and what is the version that you are using, what it depends upon, and and who is the author, who published it, license information, right, and other, okay, and some reference material for this, like if you want to know more about its usage and what it is all about, okay, any paper which is published, package is available as ADI Philo 1.1, okay, so like that, yeah, okay, depending on the version that you are using, like this version you are using, so same package is here, okay, and Windows, Mac or Linux libraries that you want to download for this, and it is available at cran.r.project.org, which is a kind of repositories for such packages. So for any package you can search like this, and this kind of informative page will appear, which will give you information about how to download it and also install it. So another thing that we do in genomic, taxonomic or metagenomic analysis, or even basically any analysis where you want to see where there are multiple dimensions and you want to visualize the complex data sets, that is the principal coordinate analysis, right? So it is a method basically that tries to reduce the dimensionality, and generally like data sets are multi-dimensional. For example, several samples with several parameters or values, so basically several dimensions are there. So it will try to reduce the dimension and make a plot, okay, so for meaningful statistical analysis, okay. So it will, while it is reducing the dimension, it is retaining the key relationship among the samples, okay, so that you can do the analysis. So it is one, so basically the outcome is a graph that tells the relationship between various samples. They could be closely related, they could be overlapping, they could be clustering together, or they could be far apart. So that will tell you the picture of the data, that how your points are separated in space. Okay, for example, this is the file, okay, right, which is in a CSV or TXT format containing 40 samples and 270 features, or basically amplicon sequence variants, or ASVs, which are basically the OTUs, okay. Earlier they were called OTUs, now they called amplicon sequence variants. So this is an example of metagenomic analysis. We will also cover it there, but just to tell the usage of R. So basically what we do here is we find the basically we cluster the sequences together. It could be any hypervariable region or 16 ribosomal RNA, which is used in the taxonomic analysis of metagenomes, okay. And after that we identify the clusters and name them as a single ASV. So there could be multiple ASVs, or basically if I say 270 ASVs, that means about 270 species or species level things, clusters have been made, right, for the various samples. So S1, S2, S3, S4 and up to S40, their samples will be there, okay. So it has several dimensions, you can see, but now when you want to plot it, you want to see them so that how these points will be separated in space, which samples are close to each other based on the amplicon sequence variants that are there, okay. So basically principal coordinate analysis, here it is with the Bray-Curtis dissimilarity matrix. So basically the objects that are very close to each other are have lesser dissimilarity, whereas which are far apart are more dissimilar, okay. And basically it will have various A1 and 2 axes and like that, which tell that how much variation you have covered in the data, okay. So basically more information can be found here. So okay, so basically if you want to use it, for example, install packages vegan library, load the library, okay, and then read the data in a, okay, so read.csv, it is in CSV format. So basically input.txt separated by /t, that means the columns or the data that is there is separated by tabs, right, which is generally like default tab or space separation is there. Tab is better because space can occur anyway, like if I write any two words, you tend to put a space in between, but tabs you put only between columns and it's a nice delimiter, okay. Then header row is present, and basically the first row is the we are using, okay. So then basically you will calculate the break dissimilarity between the samples. So okay, so break distance you want to calculate, so distance will be captured here, right. So the data matrix that you have read from there, okay, and the method that you will apply break is, okay. So this is now stored in this variable, okay. Now you will be performing the the statistical analysis. So basically for the multi-dimensional scaling, so perform principal component. So k is equal to 2, that means you want to do for 2-dimension plot you want to make, okay. So basically this will calculate the distance, right, based on the Bray-Curtis method that you have defined above, okay. And for 2-dimensional plot it will make, and it will be saved as $plot, which you can, okay, so which you can plot, okay, and get the basically you can plot it as a xy axis, and you can see that how well the points are separated. So like this was the total data, okay, and the distance between the sample that we have calculated there is now here S1 S2, basically between the sample S1 with S1, S2 with S2, because S1 and S1 are same, the distance is 0. Between S1 and S2 the distance is this. So this matrix will be actually translated into a figure like this, okay, the plot that I was mentioning there. So where you can see that there are axis 1, axis 2, right, and it is the points of are which are of the two different data sets, or okay, are like you can see that the red ones and blue ones are slightly overlapping, but you can say that majority is overlapping, which means that they are clustering together. So there is a good overlap or correlation between the two data sets. But here in this case you can see that the points are very far apart, only like few outliers are there like this or this, okay. So which means that basically the two data sets are well separated, and you can see that PC1 principal component 1 is explaining almost 71% variation, which is which tells that the result that you have got is significant and useful, okay, and you have clearly separated the points, okay. That means the two data sets were very distinct. Like in this case this is the metagenomic data from colorectal cancer patients and the healthy and the individual. So it is showing that they are well separated, and this kind of graphs are needed for publications, okay. And like this is ggplot2, another package, okay. Same way you can install it first, load the library, okay, and then add significance values, again load another library, right. And for creating customized plot with grids, load another library, okay. Now this is a data type, you have defined the status, which is healthy and diseased, okay. So there are like multiple samples, okay, 1 2 3 4 5 6, 3 are healthy, 3 are diseased, okay. And you can see that the values are calculated for each sample, okay. And then you are using the ggplot2 to basically make a box plot of the two, okay. So you want to create a box plot and also do the Wilcoxon rank sum test, okay. So basically the file is that we have just seen, this file is bacteriax.txt separated by tab. So these is separated by tab, okay, which is read in a table, fed into the data variable stored there, okay. Header is true because this is the header, right. And then you are doing the calculations, right. And then basically since there are 3 samples, so each, so I is from 1 to 3, okay. And then basically you are applying the Wilcoxon rank sum test, okay. So you have stored the data sets into disease all and healthy all, the two types of data or the status based on the status that you had, okay, and then you are applying the test, okay. So using the healthy all, diseased all, two-sided test, that means you want to go for both the positive and negative values, okay. And okay, so basically the description of the command that I am using here are here. So basically this line is performing a Wilcoxon rank term test, which I just mentioned, okay, and a two-sided test, that means it is calculating both for the possibility of differences in both the direction, that is the positive side or negative side greater or less than, okay, and it will calculate the p-value for the variables. Okay, so far in the previous slides we have looked at how to initiate the ggplot2 and also how to carry out the Wilcoxon test and save the test results in a variable Wilcoxon CI. So now we want to plot for, this is just a sample table, right? This is how your data may look like. For example, status could be healthy or disease. You might be comparing two populations or set of individuals in healthy and diseased, and some values calculated for them. It could be proportion, it could be the significance values, okay, anything that you want to plot, right? Now basically this becomes your labels which you can use on x or y axis, right? So now we want to make the plot, okay, using the results of the previously calculated Wilcoxon test, right? And we want to make a box plot, okay. So we'll use the function ggplot, where p is a variable where the output or whatever analysis this commands will do or set of functions will do will be saved there, right? So we are initiating ggplot, data 1 is our data. We want to define the aesthetics, that means that x and y axis information, that how the plot will look like and what should go on the x axis, y axis. For example, status we want to put, right? And status and name we have taken, and basically field status, that means that we will be putting. So basically in the next slide I will be explaining the different function that we have used here, right? So and also like if you have a code like this or any code, okay, because what we are showing here is only for making you familiar with R and how to use some of the commonly used codes, right? For example, this is a code for the ggplot or making a box plot, right? So the status we have taken from this table, for example. Then the y-axis we have defined, which will be species, and x-axis will be status, that will be healthy or disease, right? Then there are two functions, geom box plot and geom significance, where when we make a box plot there will be some outliers and we want to color them differently. So we will put the color, outlier color equal to black, okay. We want to give the significance or the statistical value to the plot, okay. For that we have already calculated the table using the Wilcoxon test, okay, and calculated the p-value, which we have rounded off to 2 digits here, okay, and then say print. So if you see here, it defines like what we have used earlier. So basically your command is like that, where there is an object named p, okay, which is saving the results, okay, and this is your data set, define the aesthetics, and okay. So basically in this you can see that what we have seen in the previous slide also, we have the data and defining the aesthetics, that is the x and y axis, okay. So y axis is basically the species or any label that you want to give, okay. Outliers are labeled as black. In the significance, geom significance, we are using the significance that or p-value that were calculated from the Wilcoxon test in the previous box plot and Wilcoxon slide, right? And then printing the final plot. So if I have a code like this, and if I do not know that how to know what is going on this code, and you want some detailed description of this, so I am showing you one easy way to do this is ChatGPT, where you can take this code and in the ChatGPT box you can just write this code, okay, paste, copy paste, and then say explain this code line by line, okay. So what it will do is it will process the code and tell you like what its code is intent to do, okay, what in which language or library it is using or what function it is using, and a description of, for example, p This line creates a ggplot named P using the data frame data 1 and like that. Okay, live species, adding label to the y-axis, geom plot outlier equal to black, that means outliers are colored as black. So basically what I described earlier, ggplot also give a point wise description of each and everything that was going on in the plot, and that can be very helpful when you are dealing with any code. That could be a Linux script or a Perl script or Python script. Okay, you may decide to give the entire code also, or some part of it, or a sub part, or a small part of the code, depending upon your requirement. Because if you give too long code, then also it will print a lot, right? And then you have to go through each and every line. Okay, so ChatGPT is very useful in this way. Okay, then we are executing this in the R studio. Okay, so you are pasting your code here and then running your code on the console, right? And then as we know, like it has 4 windows. So in one of the window, it is also showing you the box plot that it has created. So this is the test value, 0p equal to 0.002, right? The status, that was healthy and disease, that we have taken from the table that we used for giving the input. The bacteria species, right? Bacteria X, the number that we have given in that table, right? So basically it has more options also to change colors and showing more values here, but we are showing you just the basic of how to initiate the ggplot. Similarly, one more tool or package that you will be using in either metagenomics or in when you are doing machine learning or AI based, when you are doing feature selection, either basically it could be features, it could be in metagenomics or in machine learning also, or you want to do some prediction from the metagenomic data, right? So one of the software that you will be using is Boruta. Okay, so Boruta is based on random forest classifier, right? So which is an ensemble based classifier, and it's a kind of wrapper around this random forest classification algorithm, which deals in classification of the data. For example, if you have 6 labels, like 6 different categories, and you want to divide your data into 6 categories, or so for that purpose, random forest can first train on a set of data and then predict that a given query will be classified into which of the 6 classes, like that. Okay, so now they have used Boruta. They have developed Boruta based on random forest classifier. Okay, and this is basically used in selecting the features. So for any machine learning, basically there will be features. For example, if you see an apple, there are features like its color, weight, texture, sweetness, or shape, okay, or anything that you think may be important in identifying that apple using machine learning, that you don't tell the machine that it's an apple, but based on the features it will tell. Okay, so some features may be more important, some features may be less important. Okay, so that is why, and this is just one example, but sometimes you might be dealing with situations where there are thousands of features, like it could be descriptors or fingerprints in case of like structures of molecules, right? So there, picking the important features is very important. Otherwise, and basically it is also important because those features are actually playing a role in identification or classification of that object or molecule or any thing, any input query, right? So first of all, why to waste time in dealing with features that are not relevant or not contributing to your calculations? Okay, it will also save time, and it will be much faster and better, right? The final code that you will make or the model that you will make. Okay, so Boruta is one package which was published in 2010 and has been updated later. Okay, which is convenient to use also. So to install, basically you can directly say install.packages Boruta. Okay, then load the library Boruta. Okay, then basically when it is doing the calculations, okay, so it can start randomly from any of the feature. Okay, so if you define a seed, for example, randomly the seed number like 42 has been defined. So this is generally to do that you get reproducible result every time you run this software, right, on the same data set. Otherwise you may get slightly different or like significantly different results also every time. So setting up the seed is a good idea, and that is why you may say set seed as 42. Then read the data from your file in your system, like read table file, the file name was input.txt containing an header, so header equal to true. The separator or delimiter that was there in the file, it was say column file, and columns are separated by tabs, so \t. It could be \h also if it is separated by space or comma or like that. Okay, row name equal to 1, that means you are dealing with the header row. Okay, then since you have two columns here, so basically you will define that to what column it should read. The p-value, right, the Monte Carlo adjustment is set as true, maximum iterations it should do is 100, tracing 0, that means it will not print information on the screen. If you otherwise it may put a lot of descriptive information to know that the status of the process which is going on the background, right. Hold history equal to true, and get important function, that means the significance, right. So basically this we will also be doing in the next slide, that what is the description of this. And also again for this code I will show how ChatGPT can be used to reveal more about the code. Okay. Then finally you will be first training and then writing the final decisions and tables, right, in write.table, where you will be training on the final decision file. Okay, and okay, separator is quote is equal to false, and okay, then Boruta importance is you want to calculate, okay, which will be given also as the output. So here you can see that basically in ChatGPT when I pasted this code, right, so it also analyzed the code and gave you all the information, like what columns were used here, okay, and the status and p-value, what was considered, okay, max runs, do trace, hold history, right. So everything that I described in the previous slide, ChatGPT also gave a descriptive information about the code. Okay, and basically this function get IMPRF, so it is a method to compute feature importance, because that is the purpose of running Boruta also, and using the random forest algorithm. Okay, so this is the code that we just ran, okay, and the description of that we have shown by ChatGPT also, and in this slide. Okay, we have already covered this in the previous, so I am not going into detail of this again, right. So when we are running this code on the console, it will check the various variables and tell that which variables are basically selected, or which features are selected or confirmed, right, and others it has rejected. So this way it can select features, and among all the possible features it is calculating or selecting those features which are mattering in your final decision, or identifying something, or whatever the problem you are dealing with, right, where the feature selection will lead to the next steps of the model construction. Okay, so basically in this lecture we have looked at what is R, its installation, how to use it using RStudio and console, and some of the commonly used codes, tools and packages that are used in R. Okay, and this is very, very important for the genomic analysis, metagenomic analysis, machine learning, right. Generally a combination of Python and R is very much recommended, because Python is becoming the language of choice for machine learning also, right. And with this lecture and the previous two lectures, where in the the first lecture in this week we looked at the Linux, right, and then in the next lecture we looked at how to execute the Linux commands and setting up files on the Linux system, right. And along with R, basically these three things will help you to prepare your system and get familiar with the tools, packages, and the installation generally is similar either for R or Linux, right. And because generally the same type of installation procedure is used by different developers, right. So all these three will help you in running the codes or program that we will be looking at in the later part of the lectures, including the genomic analysis or metagenomic analysis or epigenomics, right. So I suggest that along with these three lectures, you should also read books, and you can also search codes on Google which can help you in sometimes when you don't want to rewrite a simple code which is already available or written, right. GitHub can also provide you a lot of codes, right. So the books that I recommended, generally O'Reilly publications, either R, Linux, or Perl or Python, can be taken from there. The e-versions are also available. So in the next lectures, now we will be going forward in the details of the genomic analysis. So this is good for now. Okay, thank you.